Hi, everyone, and welcome to my talk. My name is Tawny Reapy. I'm from the Colorado State University, Colorado Cooperative Fish and Wildlife Research Unit, and I'm going to talk to you today about comparison of tissues for the detection of Renibacterium Salmoninarum in cutthroat trout of Colorado. First, I'd like to thank the co-authors on this, Dr. Eric Featherman from Colorado Parks and Wildlife and Dr. Dana Winkleman from Colorado State University and the Colorado Cooperative Fish and Wildlife Research Unit and U.S. Geological Survey. So bacterial kidney disease is a disease associated with salmonids across the nation and across the world, decimating fish and fisheries and aquaculture facilities. Due to these high mortalities among fish populations, bacterial kidney disease has been an important focal point for many years, and how to test for the pathogen leads to many of our research objectives. So Renibacterium Salmoninarum is the bacteria that causes the bacterial kidney disease, which I'm going to call RSAL for short from here on out. And RSAL is a regulated pathogen in the United States within hatcheries and aquaculture facilities and can be found in salmonids across the U.S. And this map is representing RSAL prevalence data in salmonids from the U.S. Fish and Wildlife Service Wild Fish Survey from 1996 to 2015. And we can note high prevalent sites in this darker purple color, which can be seen in the Pacific Northwest in Chinook and coho salmon, in the Midwest in brook and brown trout, in the mountain ranges of Montana, Idaho, and now Colorado among rainbow and cutthroat trout. Specifically in Colorado, Colorado Parks and Wildlife and the U.S. Fish and Wildlife Service conducted a survey to determine the prevalence and distribution of RSAL in Colorado fisheries in 2017 and concluded that RSAL is pretty widespread throughout the state, but bacterial loads are low and clinical disease is often rare. Moreover, there have been scant detections of RSAL in Colorado hatcheries since the 1960s, with a few in the 90s, but till recently, 6 hatcheries, 5 state and 1 national hatchery have tested positive for the detection of RSAL. Specifically, in cutthroat trout reared in Colorado hatcheries, infection intensity, or the loads of bacteria per individual, has been low, but prevalence has varied from low to high. However, due to the nature of bacterial kidney disease, some environmental stressors may contribute to disease or high mortality events among infected fish, which is one of the reasons I mentioned that RSAL is regulated. Currently, there is no treatment for bacterial kidney disease or RSAL infections, but there have been some methods to limit transmission, including chemotherapy and vaccination, which include erythromycin injections. But because there is no treatment, detection is key to preventing RSAL infections in hatcheries. So we need to make sure that we have sensitive and specific assays both in the laboratory and with our tissue collection methods. The AFS Fish Health Blue Book states that you can detect RSAL with a presumptive testing method with DFAT or with ELISA and confirm a positive infection in the tissue with confirmatory testing with either selective kidney media for culture or PCR assays. And it's important to note that if the presumptive test does not detect the presence of RSAL, then the confirmatory test is not typically completed. But if the presumptive test is positive, then the confirmatory test is completed, which could lead to implications with management if the presumptive test is not highly specific or sensitive to detecting RSAL. And so since DFAT is what we are using to screen most fish for RSAL infections, for that presumptive testing method during fish health diagnostics and assessments in hatcheries, we should be able to assume at least 90 to 100% detection probabilities with, again, this presumptive testing method. Furthermore, current tissues that you can collect for RSAL testing based on the AFS Fish Health Blue Book include lethal testing of kidney tissues, and newly added are other hematopoietic tissues such as liver and spleen. AFS also states that you can collect ovarian fluid, but of course this is going to be limited only to adult spawning females. And similar to diagnostic assays, we need to make sure that we're collecting the correct tissue in our cutthroat trout in Colorado to capture RSAL infections in the tissues. Which brings us to our study objectives, which are to determine the best tissue to use for RSAL detection and determine the best assay to use for RSAL detection in Colorado cutthroat trout. So in 2019, we collected 781 adult spawning cutthroat trout and collected 6 tissues from each fish. For our primary objective, we sought to determine best tissues to use for testing for both lethal and non-lethal study objectives, and collected 3 lethal tissues, liver, spleen, and kidney, and 3 non-lethal, which include blood, ovarian fluid, and mucus. However, for this talk, we're only going to focus on the kidney and liver tissues. So tissues were tested with both assays of DFAT and qPCR for our study. So we did not follow the typical methods of the presumptive, then the confirmatory testing. We just did both. And also each tissue was tested twice for each assay, which we defined by a survey. Therefore, we have 2 surveys for each tissue and for each assay. So our results indicate that out of 781 total fish, there were 25 fish testing positive with DFAT when we tested the kidney tissue, and 267 fish with kidney tissue tested positive by qPCR. Then looking at how many fish tested positive with liver tissue, 58 fish were positive by DFAT and 448 fish were positive by qPCR. So clearly, just with these raw numbers, we can see more fish test positive with qPCR and not with the DFAT method, which again is that presumptive testing method. So I would like to point out, we do have an assumption within these methods that if a result of an assay was positive, there was a true positive test. And while we can have a true negative test result, there may be false negative results, which we predict is probably occurring with these DFAT results more so than with the qPCR, and arise when the assay does not correctly identify the presence of R cell in the tissue. And since we're predicting these high possibilities of false negative results, we need to determine the detection probability of both assays we're using, especially with the presumptive testing method. So using this estimate of detection probability, we can then understand the true underlying occupancy of R cell within different tissues we may collect to test, such as the kidney and the liver, which bring us to an occupancy model. So typically in a traditional occupancy model, there's some underlying biological reality of occupancy that we do not know at a site. And so our site is defined as an individual fish. Then we try to detect or observe the presence of the species among the patches in a site. In our case, R cell among different tissues, which is the patches in the fish, again, which is the site. And so we do this with some detection process. Here we have qPCR and DFAT, which then gives rise to our presence and absence data of 1s and 0s, and this is completed with at least 2 or more surveys. Our detection data could then be a combination of detection and non-detection data in the form of 1, 0, 1, 1, 0, 1, and 0, 0. But the problems really arise when we have detection data of 0, 0, when the bacteria is actually present in the tissue, but we just cannot detect it with our current methods. So then to expand on this detection process with multiple tissues are infected, we go to a multi-state model, where instead of having our response from our observation process be solely 1s and 0s, we have 4 states of infection as our response, which are labeled as 1 through 4. Then we have the true states, which is this underlying biological reality again, so the true state of R cell infection within the tissues, which in each fish. And we then have the observed states, which arise from our detection process by the DFAT or qPCR. So the first state is when kidney and liver tissue are both negative by an assay. An example of this detection data from what is observed may look like the following, where on each tissue, kidney and liver, we conduct 2 surveys or tests from one assay, either qPCR or DFAT. From the first survey of the kidney, we do not detect R cell, so it gives us a 0. On the second survey, we also do not detect R cell. On the first survey of the liver, we did not detect R cell, and so we get a 0. And again, on the second survey, we do not detect R cell. And that is what makes up the first state of infection in terms of what our data looks like. The second state of infection is when a kidney is positive and liver is negative, and one possible combination of observed detections may look like this between the 2 surveys. The third state of infection is when kidney is negative and liver is positive, and detections may look like this for one of those possibilities of our detection data. And the fourth state of infection is when both kidney and liver tissues are positive, which is noted by all ones across both tissues and all surveys. So not only can we observe a fish in the true state of infection, but because of the possibilities of false negative results, our observed state may not be the true state of infection because our assays did not detect the bacteria in the tissue correctly. For instance, if we observe a fish with a negative kidney and negative liver, we may actually be missing the infection, and the true state could be either combination of kidney or liver tissue that is actually positive. This is similar for the other states as well, where we can observe the true state of infection where kidney is positive and liver is negative, but the negative liver may be a false negative, and therefore the truth is actually state 4, where they're both positive. Same is true for state 3, where we can detect the truth of kidney negative and liver negative, but the kidney could be a false negative, and the true state of infection may be kidney positive and liver positive. But with the last state, if we observe a fish with both kidney and liver tissue positive, we assume that it is the true state since we are not accounting for any false positives in our model. So we have these multiple possibilities we account for to estimate our probability of detection and occupancy in each state, which can then be translated into these probabilities of the state of RSL infection for each individual fish, which we then use to fit a Bayesian occupancy model with vague priors to determine detection probability for the 2 assays and occupancy of RSL among each infection state. So from our models, we can estimate detection probability for both assays and across both tissues, derived from detections from those replicated surveys with each tissue. And so this plot is a posterior density plot with qPCR on top and DFAT on the bottom. The DFAT is not any negative results, it's just a mirrored plot with detection probability on the x-axis. And we can see qPCR is better overall with these higher peaks with detection probability of kidney around 73% and liver around 61%. We can see DFAT is much lower with a detection probability of kidney around 54% and liver around 33%. We also see much less variation by the little width and higher peaks with qPCR than DFAT, further indicating that qPCR may be the best method for detection between the 2 assays. And so using these estimates of detection probability, which again is the estimate between the 2 surveys, we can use these to look at the conditional probability of detection. So these conditional probabilities are a function of the estimated probability of detection from our occupancy model, which is then conditioned on the probability of occupancy given the infection states when kidney is positive or when liver is positive. So when we look at these conditional probabilities, the probability on the y-axis and the number of surveys completed on the x-axis, we can see clearly that the with DFAT detecting a liver infected with RSL, we have much greater probability of detection than with the kidney tissues. And we see this again for qPCR, where detection of RSL in liver tissue is much greater than kidney tissue. Most notably, when we think about the number of surveys actually conducted during routine fish health assessments, where we actually only conduct 1 survey. So we can see that with 1 survey, our conditional probability of detection is reduced to 32% with liver and 21% with kidney for DFAT. Then with qPCR with 1 survey, our conditional probability of detection is 52% with liver and 34% with kidney. So overall, not only does using DFAT for our testing method and kidney tissues as our tissue collection type reduce the conditional probability of detection, but with the lack of multiple surveys completed on each tissue with each assay, it also reduces our detection probabilities. Now, if I still have not convinced you that the DFAT method is not very good if you do not do replicate surveys so many times, let's look at the number of individual fish that was estimated to be in the state of infection of 1, where both tissues are negative, and compare these numbers across DFAT and qPCR from our occupancy model. So here, 652 fish are estimated to be negative by DFAT, and 159 fish were estimated to be negative by qPCR. So we have an approximately 493 fish that were determined as negatives by DFAT, but are actually a false negative result by DFAT. So if we were conducting a traditional fish health assessment where we need a presumptive positive test to conduct that confirmatory test, we would never test these 493 fish with qPCR because DFAT was negative. And this can be a huge issue in terms of management and movement of fish after routine testing for arsal infections among hatchery fish because the fish are considered negative, and so movement of stocking of fish is permitted, even though they might actually be infected. So our conclusions then overall is that liver tissue is best for testing. However, a combined effort of collecting kidney and liver tissue and testing them together may be optimal. We also note that detection probability is not 90 to 100% for DFAT or qPCR, especially with only one survey that is typically done during routine fish health assessments. And so using DFAT as a presumptive test could lead to high false negative results. And so we suggest not using DFAT alone for presumptive tests because, again, this could lead to transfer of infected fish or eggs throughout the state by stocking when we have a false negative result. We also suggest that if other testing is not available to test individuals, maybe we actually test with higher number of replicate surveys on the tissues 5 or more times with presumptive and confirmatory testing methods. And so I would like to thank a lot of people at Colorado Parks and Wildlife and Colorado State University, as well as my committee members that helped on this project and all of our funding agencies. So thank you again for listening.